Hi everyone, my name is Mingming Cui. Today I'm going to talk about some new work that I have been working on Gene Environment Association to identify SNPs associated with climate in Asian longhorn beetle using pooled whole genome sequencing approach. The Asian longhorn beetle, often abbreviated as ALB, is an invasive species which is native to China and South Korea and has introduced to North America and Europe for decades. It mostly feeds on poplar, willow, elm, and maple in its native range, while in North America, ALB preferentially feed on maple trees. Usually it has one generation per year. Adults dig pits in the bark to lay eggs. The young larvae feed on the cambium area and later tunnel deep into the vellum. When they become adults, they emerge by chewing around exist holes around the bark. The draft genome of ALB has been published and it contains thousands of scaffolds. One of the most important reasons for ALB's success as an invasive pest is its adaptation to cold. For many insects, extreme temperature is a matter of life or death. Insects have to develop strategies in order to survive temperature extremes. ALB larvae are known to be freeze tolerant, potentially thanks to their increased production of sugar contents such as glycerol during chilling. There is a wide range of climate differences among the regions of ALB distributions, which across several climate zones in China and Korea. ALB adults cannot survive cold winter temperatures, but their larvae can overwinter and survive under cold temperature. It can be assumed that ALB developed specific physiological strategies to resist low winter temperatures in temperate climates. We are interested to uncover genomic signatures underlying adaptation to temperature. Therefore, with the Genome Canada-funded BioSafe project, we collected global populations of ALB, sequenced their genomes, and applied genetic and computational approaches to develop genomic-based biosurveillance tool for this species. By combining information from phylogenomics, population clustering methods, simulations, as well as genotype-phenotype associations, the BioSafe project identified genetic markers useful for the development of genomic biosurveillance tools to identify species, assign source populations, and predict risks of spread. With that being said, one of our goals is to identify genomic signatures of cold tolerance to help us answer important questions in the future about the adaptive potential of intercepted ALB specimen. In this project, we have already identified population structure of native ALB populations earlier. The work has yet been submitted to scientific journal. For native ALB populations, we perform sampling in the native range as shown on the left map within 20 sites. In China, our sampling covered major geographical regions as color-coded on the map. Within China, the discriminant analysis of principal components helped identify 6 genetically distinct clusters among ALB populations. Now that to determine genetic variant that associate with cold tolerance, we sequenced the whole genome of 10 pooled populations. We set up a Pool-seq experiment that grouped samples within the pools based on 2 criteria. Firstly is the similarity of population structure as identified through the GBS and structure analysis previously, and also the similarity of the climate. We mixed equal amounts of DNA from 20 samples for each pool for a total of 10 sequenced pools. We performed novel-seq parent sequencing. The raw reads underwent QC and read alignment, and we caught variants using GATK and SAM tools. After variant calling and filtering, we performed the 2 GEA methods to detect climate-associated variants, redundancy analysis, and bypass. To evaluate the accuracy of allele frequencies obtained from the Pool-seq data, we looked back into the individual genotype GBS data that we used in the population structure analysis. On the left figure, it shows the correlation of allele frequencies from Pool-seq and GBS. On the right figure, it shows the allele frequency correlation distribution between GBS and the Pool-seq. Basically, the allele frequency obtained from the two approaches are quite similar. Then we used a redundancy analysis, which based on a linear model to detect loci under selection. It assumes a linear dependency between the response variables and the explanatory variables. So we kept the environmental variables downloaded from WorldClimb with a correlation coefficient less than 0.7 and performed RDA analysis with 4 variables, that is isothermality, the minimum temperature of the coldest month, and also 2 precipitation variables. The results show the first 2 RDA components explained more than 65% of the variance, and we identified 224 outliers that significantly associated with the climate variables, and the minimum temperature of coldest month associated with the majority of the outliers. For the 224 candidate loci associated with climate variables, we attempted annotations by running blasts against all databases. We used the flanking 2 kb nucleotide sequences to do the blast. Overall, we found only a few loci annotated by geoterms or CAG gene database. And secondly, we performed Bayesian hierarchical model-based method bypass. It calculates this XTX statistic, which can be viewed as a SNP-specific FST explicitly corrected for the scaled covariance of population allele frequencies. The strength of association is given in the test by estimated Bayes factor. SNPs with Bayes factor more than 20 dp is treated to be a candidate for selection with respect to a given covariable, which is temperature in this case. With that, we identified 24 SNPs that are strongly related to the temperature covariate. We also annotated the candidates, only 10 out of 24 with annotation against NR database. To conclude, it has been proved Pool-seq is an cost-efficient and also robust approach in terms of generating accurate allele frequencies of massive SNP markers across the genome. And with this sequencing approach plus the computational methods, we identified 248 promising candidates. Although the functional annotation of the candidates need to be improved in order to better understand our candidate genes. And hopefully our findings will be helpful for the ongoing genomics-enabled biosurveillance tools development. And And I would like to thank my supervisors, my co-workers, and our collaborators, and also thanks to our founders. If you want to learn much more about other aspects of the Pool-seq project, please visit the website. And if you have any questions, please don't hesitate to write to me. Thanks very much.